can and can't you tell just by looking at a gel? How about a blot? What can it tell you? What can it not? So today, how to get the most but not more out of your gels and blots, what they can and cannot tell you.
Much more in other posts, but in gel electrophoresis, we take a mixture of molecules, so different proteins or different nucleic acids, so DNA or RNA, and we separate those different pieces by their size by sending them traveling through a gel mesh. In general, the bigger things are going to get tangled up more in the mesh and thus travel slower. And so if you were to use like an all protein dye or an all nucleic acid dye, in order to visualize where these different molecules are located in the gel, you find that the bigger things are higher up, the smaller things are lower down. The strength of the band is going to tell you about the abundance of those various components, and the number of bands is going to tell you about basically a measure of the purity, how many different things are in the mixture. But there's a lot of different caveats about these we'll get into. And one of the most important things is that with these methods, you're not being able to tell apart, although you can tell that there are multiple things are present, or even if there's just one thing that appears to be present, you can't tell what that thing actually is. If you have an idea of what it is, then you can do some sort of blot. This can be, in the case of proteins, we're typically talking about a Western blot. And in the case of nucleic acids, we can be talking about like a northern blot or a southern blot. In these cases, we take a probe, so an antibody in the case of a protein, or a labeled matching oligonucleotide, so like a short piece of DNA or RNA that's complementary to the sequence that you're looking for. And then after you do a blot step where you transfer the molecules out of the gel and onto a membrane, then you can actually probe that membrane to see if a specific component is present. But importantly, with these blot methods, you're only being able to detect the molecule that you're looking for. So you have to have a probe that's specific to that molecule. And then if that molecule is there, you get a signal. If it's not there, you won't get a signal. The strength of the signal is going to tell you about how much of it is there, but you're not being able to learn anything about what else might be there. So generally speaking, we can say that gels can tell you about what cannot tell you about what is in a sample, but blots can't tell you about what else is in the sample. So the gels are going to give you a broader picture of how many things are in there and how much of various things are in there, and not the identity of those things. Whereas a blot can tell you if a specific thing is present and the relative amount, but not whether anything else is present. So they both have different strengths and weaknesses. And even within these general concepts, we have a lot of exceptions. And so today I wanted to go into some of these exceptions to help you get the most out of your experiments, but not to overinterpret them. So what can and can't you learn from one of these experiments? So let's go back to, we'll start with the gels. So as I mentioned, we start by sending the molecules traveling through these gel meshes. Much more on this in other posts. When we do this, the molecules are going to be invisible. If you see some sort of dye in your gel, that's just going to be a tracking dye. It's not actually bound to the molecules of interest. It's just showing you where the where the front of the gel is traveling. So you know when to stop the run before your proteins or your nucleic acids come out. And then in order to visualize them, you have to use some sort of stain. So typically we can use like an all protein stain in the case of protein gels. So for protein gels, we're typically using like SDS page. So these these vertical gels that are these thin gels. We also use page gels for like small RNA or DNA. In this case, often urea page gels. And then for like DNA or bigger RNA, we often use these agarose gels, which are these horizontal slab gels. Just a technical note, and much more on all of these and other posts. But the basic idea is the same, right? You're sending molecules traveling through this mesh using electricity and getting them tangled up. To visualize the proteins, we often use a chromacy-based stain. It's going to be either classical chromacy, where you have to like fix and defix and destain, or fix and destain, stain and destain. or these like these colloidal methods, which are simpler, more environmentally friendly because they're not using all of the methanol and stuff. It's a lot easier. You don't have to destain them and things like this. So this is really helpful, but they're both based on the same principle where you have this dye that's going to bind onto proteins and show them to you. For nucleic acids, we're often using some sort of fluorescent dye that's going to bind to nucleic acids and show them to you. Okay, so if you were to use one of these all protein stains, you'd be able to see a band for all the proteins, and then as we'll talk about later, if you were to do some sort of probing, then you're only going to see if a specific protein is there. So if I was looking for whether this protein is here, well, here you can see that I'm going to see bands if the protein is there, but not if it's not. Or at least you hope so. We'll get more into this later about how antibodies can have various amounts of specificity. So you might see bands that you think are your thing, but aren't really. In general, though, one of the things that we can learn from a gel is whether the size. So one way you can tell if that band is likely what you're looking for is where it's located size-wise compared to a ladder. So one of the things that gels are really useful for is that they can tell you about the relative sizes, or rough estimates of the sizes of various molecules in a mixture or a single molecule in a solution. Because what happens is when you're running the gel, the longer molecules are going to get traveled up more. And then when you turn off the electricity, they stop having the drive to move through the gel. The bigger ones are going to thus be higher up in the gel, and the smaller ones are going to be lower down. Because how far down or how high up they are is going to depend on things like how long you run the gel and how meshy the gel is. So you can alter the gel meshiness by using like higher percentage gel. It's going to be a tighter mesh, and therefore it's going to be better for separating the little things. And the looser mesh is going to be better for separating the bigger things. And the meshiness is going to influence how things run in the gel, as is how long you run the electricity. If you run the electricity forever, then the molecules are just going to come all the way out of the gel. If you weren't to run it very long, they would be, even the smaller guys would still be higher up. So we need something to compare it to, and this is where the molecular weight markers of these gel ladders come in. These are a mixture of molecules of known size that you can run alongside your sample in order to then use them as a sort of ruler to estimate the size of your molecules. And you can see that unlike a normal ruler, the spacing of these is not like evenly set apart. So you always have to be running one of these ladders and making sure that the molecules that you're interested in are in the range of the ladders, which is more of an issue for the nucleic acids than the proteins, because proteins tend to be all in a similar range of sizes, roughly speaking. Whereas with nucleic acids, you can have really tiny things or really, really big things. And so you want to choose your ladder accordingly. But in these cases, you would expect your protein to be located between the size of the bands and the ladder that it corresponds to. So if you're studying, say, a 90 kilodalton protein, you would expect it to be between the 75 and the 100 kilodalton lines. So with protein sizes, we're typically talking in terms of kilodaltons, which is a measure of like 1,000 grams per mole, much more on this in another post, but a quick estimate is that the average amino acid is about 100 daltons or 0.1 kilodaltons. So if you have an 1,000 amino acid long protein, that would have a molecular weight of about 100 kilodaltons. So if we're talking about a 90 kilodalton protein, that would have about 900 amino acid letters, and much more on that in other posts. Going back to our words of caution, these are not always, your protein might run a little higher or lower than you would expect for various reasons. One is that they could have post-translational modifications. So things like phosphorylation are going to add mass to that calculated molecular weight and potentially make your protein run a little bit higher up than you would think it would be. Additionally, the composition of the amino acids can influence how it runs. Different amino acids have different sizes, charges, shapes, and these can influence how the protein sample is going to run. So even proteins with the same molecular weight can have, say, different numbers of amino acids, or they can have different properties that will then cause them to run slightly different on a gel. So if your protein isn't running exactly where you would expect based on the molecular weight standards, don't freak out. But it can be really useful for things when you're trying to get an idea about the sizes. One thing that's really useful is if you are trying to determine how much of a protease to add, you say cut off an affinity tag. There's so much more on this in other posts, but when we're doing protein purification of recombinant proteins. So basically where we take genetic instructions for making a protein and we stick them into cells, just bacterial cells, and get those cells to make the protein for us. Because we're sticking in the instructions, we can actually add a little extra onto those instructions, get the cells to add a few extra amino acids onto the end that can serve as an affinity tag. It's going to be something like a his tag, so a string of histidines that can bind to metal on a resin, such as nickel affinity chromatography, or something like a strep tag, which will bind to the streptakin resin. So various ways that are going to basically provide a little handle that you can use to help purify the protein. Using chromatography, so where we have these columns filled with these little beads with something that's going to specifically bind to that tag, we can get our protein with the tag to stay stuck, wash everything else off, and then elute or push off our protein, such as adding a competitor that mimics the tag. Once we're done with that, we often want to cut that tag off. So we typically design these with a protease recognition site. So a site that's going to be recognized by a specific protein cutter, such as maybe TEV, that's going to cut off that tag. We need to figure out how optimize how much of that protease to add, so how much of that cutter in order to cut this off and how long to use it for. So for example, we might need to do a time course in dilution series to figure out the best amount. You can see that if you don't add any protease, you have a protein that's going to be higher up in size. When you add a protease, you're going to get a lower down product because it's now smaller. And you also see the addition of products you didn't see before. So you'll see both the protease and the cut product. So this is just comparing two different preparations. So this is from my old lab when I did a new purification of this TEV, and I wanted to see, compare mine to the old one in order to know if I needed to add the same amount, more or less, etc. And so you can see that the more of the protease you add, the more of the cleaved product that you get. And so then you could use this to figure out how much to add. But as I mentioned, there can be caveats. So in the case of the proteins, we talked about how different composition can affect things. One of the biggest things that can affect how things run in terms of a nucleic acid gel is the relative topology of the molecules. This comes into play a lot with plasmids. So if you have a circular piece of DNA, if you have a linearized piece of DNA, it would run at the size you would expect it to. There can be some slight differences based on the composition, but because you only have those 4 nucleic acids, C, A, T, and G, and they're going to have less of the variability based on composition, as opposed to proteins where you have 20 amino acids and those all have very, very different properties. But what you can get with DNA is that, especially if you have a circular piece of DNA, what's going to happen is that because it's not linear, it's not going to travel just like it was a straight line. And so you're going to have effects based on whether it's all circularized, whether it's nicked, so one strand is cut, whether it's really, really coiled, so it's super coiled, you're twisting a rubber band and keep twisting it and it'll get all crunchy up. Then it's going to travel faster, called the super coiled. And if it's nicked, it's going to travel even slower because now it's kind of like this weird shape, and now you have something sticking out that's going to cause it to drag. versus when it's linearized. So when it's super cold, it's going to travel faster than you would expect. When it's thick, it's going to travel slower. So all these things can influence how fast the protein runs, or I mean, the nucleic acid runs. Okay, another thing gels are really good for is telling you about the purity. It's because each thing that's in there is going to represent, in theory at least, a different product. And so you'll get each of these different, this is a protein gel, so each of these bands would represent a different protein that's present in the mixture. And say maybe you want to compare protein preparations of different proteins that you're purifying, maybe different versions of the same protein. So in my graduate work, I was working on this protein arginine, and I was doing a lot of different mutated versions of it to test the importance of different amino acids and things like that. And so I would have a ton of different versions, and I wanted to make sure that I was loading equal amounts of each of these proteins and that they were relatively similar purity. You can also use this idea of multiple bands being an indicator of purity when you're doing some sort of purification regimen. So I talked a little about chromatography before, but in the sense of affinity chromatography, but there are other types of chromatography we can use as well, such as ion exchange chromatography, where we're separating proteins based on their charge, bits with charged resin, so the charged little beads, and size exclusion chromatography, where we're separating proteins based on their size. And we often use these, a combination of these in order to get proteins purer and purer and purer. So here's an example from undergrad or grad school where I was talking about that protein I was purifying. So I purified it with a sumo strep tag. So with that strep tag I was talking about, we're running gel now. I see this product that's high up. If I do tab cleavage, you can see that the product moves down, and you see the addition of additional products as we have a band for the tabs that we added, as well as a band for the cleavage product. I can then use cation exchange chromatography, so separate molecules based on their size, followed by gel filtration. So followed by that size exclusion. Gel filtration is another term we use for it. And now you can see that I'm removing these other products, leaving me with a more pure protein. Also in purification, we can use use gels in order to determine which of the fractions to keep. So for example, when we're running size exclusion chromatography, how it works is that these beads are made up of these little, these beads have these pores in them, and the smaller things are going to travel through all the pores, where the bigger things are going to get excluded from the pores. Because the bigger things can't go through all those little secret tunnels, they're going to get to take shortcuts. They don't have to go as far. And so they're going to travel faster and come out sooner. So this is the opposite of what you would see with a gel. But then what's going to happen is that then the smaller things are going to come out later. And as they come out, you're collecting them with a fraction collector. So you're separating, like say you have a mill, then the next mill you go into another well, the next mill you go into another well. You can then, this is typically going through a UV detector, so you can see that the protein, because proteins absorb UV light, I believe my are 280 nanometers. We can then detect when proteins are coming off, but we can't actually see those proteins. We can't see how pure they are. We can't see what size they are, although we can get an indication that the bigger things are going to be coming off before the smaller things. But often these are traveling together, and so you could have a mixture. And so before you go and you pull together the fractions that have your protein of interest, so you can then concentrate them and work with them, you want to make sure that you're only you're collecting the right fractions, and that those fractions are, and if you want them super pure, you might be actually taking only a subset of what's actually there, because you want to choose the purest fractions. So you can run them on a gel, and you can see that some of these fractions, if I wanted super duper pure protein, assuming I was going for this protein, then I would take just these fractions. But if I wanted to get like the most bang for my buck, I would be collecting all of these fractions. You can see that in the earlier fractions, I'll get more of the bigger things, and the later fractions, now I see more of the contaminants from things like the TED and like the tag. So I talked about how the number of bands is going to tell you about how many things are present in the relative purity. Well, there are a lot of caveats, and one is that what you can and can't see depends on how much you load. So if I load a ton of this protein, you can see that it looks pretty impure. But look how much of the main protein there is. This is like so much more than these little things. So if I were to run a much smaller amount, you probably wouldn't even see that those little other bands were present. Also, if you overload your lanes, so if you load too much, you kind of just see a smear and you can't even make out the individual bands. So you need to be optimizing how much you add. Remember that if you're working with a purified protein, then the total amount of protein that you want to add is going to be lower than the amount you would add if it was not a purified protein, because often it's telling you how much to add based on the like the total protein amount, but that's spread up over all the different bands. If you have a single band, well, now you can more easily overload it. So typically I would say somewhere like 500 nanograms to 1 microgram of a specific purified protein would be what I would add. If you have a big band, it can easily drown out multiple bands. Even if you have smaller bands, if they're similar in size, you can't tell if there's one band present or two or three or four or five or six or seven, because they'll all be on top of each other. And so you might think that you have a pretty pure protein, but there's actually multiple things there. If you want to get a better sense of how many things are there, one thing that you can do is load less so that you have less risk of the bands overlapping one another. But then you have the problem of potentially not being able to detect the lower amounts of things. So you can do a more sensitive method such as silver staining, where you're then able to see the smaller amounts of contaminants. But even this has a sensitivity limit. So because you're starting with less as well, you might not be able to see those additional products. One thing that you can do to separate bands that might be running close together is to make sure that you're using a gel with the optimal resolving range. So as I mentioned before, you can change the meshiness of gels in order to kind of control whether it's better for smaller things or better for bigger things. So you can think if you have really big holes, all the small things are going to just travel easily through those holes, no matter if once they're below a certain size, they're not going to separate out better, because it's like if you had just giant holes, and anything that's smaller than the hole is just going to go right through, no matter if they're a lot smaller or a little bit smaller. But you would have better separation of the bigger things. But oppositely speaking, if you have really small holes, then all the bigger things are just going to either not go through, or they're not going to separate out well. So we need to choose a concentration of gel that's going to be ideal for the size of the molecules you're interested in. As you can see, based on this migration chart for a ladder in these different gel types, depending on what type of what percentage gel you have, you're going to get better separation of the smaller or bigger products. So for example, if you used a lower percentage, so like a 7.5%, you'll get better separation of the smaller things. But if you use if you use a lower percent, you'll get better separation of the bigger things. If you use this higher percent, you're going to get better separation of the smaller things, but worse separation of the bigger things. If you only have like a single protein that you're interested in, then this is easier. If you have a range of proteins that you're interested in, you often need some sort of gradient gel, but it can be harder to optimize for the protein that you're interested in. Another thing that gels can tell you is based on you can't tell the identity of the contaminant bands, but you can get a sense of whether these bands are indicating that your product is getting degraded. So as I mentioned, you can't tell the exact identity of the molecule, but if you're purifying a protein, say, and especially if you've done some sort of validation step to make sure, maybe you do a blot, where you do mass spectrometry to test the identity of the band, you know that that band is really what you're looking for. Then you can say, okay, well, are the little bands underneath that product that could be representing degradation of my product? So in addition to having those other bands that can represent different proteins, you can also have, or different nucleic acids and nucleic acid, you can also have bands that are representing degraded versions of your protein or your nucleic acid of interest. And these can come about because there are molecules like exonucleases and endonucleases. So nucleases are DNA and RNA chewers, as well as proteases, which are protein chewers. And so these can cause degradation of your products. Can be a really big deal for nucleic acids, especially RNA, because there's like this RNase A, which is a type of RNA, a really robust nuclease that can degrade RNA. And so here you can see they were testing the ability of an autoclave to destroy RNAs. But the basic idea here is that if you add this RNase, especially with autoclaving, so autoclaving can't kill it, it can reduce a little. What you can see is that with a lot of RNAs, you're going to get a lot of this degradation. So you see these small products underneath that main thing of interest. So even if you start with a relatively pure product, you're going to see all of these smears and smaller products beneath it. This can be especially evident if you're using some sort of radio labeling. So you use radioactive phosphate group to the end of these products. And this is going to allow you to track them. And so here you can actually get, because this method is so sensitive, you can see degradation products like this that you might not be able to see with just like a urea page or some other fluorescent stain. But when you see degradation products, you often see them as smears or individual bands underneath the major products of interest. Another thing you can tell when you're doing labeling is whether you have excess of the free ATP present, which you might see as like a smaller band lower down, indicating that you weren't able to get rid of all of that free ATP with your post-labeling cleanup. And so much more on this in other posts. With proteins, you also have the issue where you could see degradation products that are going to show up as smaller little bands underneath your bigger band. And if you see something like this, it might indicate that you want to be adding protease inhibitors or nuclease inhibitors in the case of DNA or RNA. And you want to be working more carefully, maybe keeping your samples on ice, avoiding freeze-thaws, working with them when they're fresh, and that sort of thing. We talked about how we have the size and we have the purity. And when we're talking about purity, this is assuming that you kind of know what the molecule is. And so as I mentioned, you can't tell the exact identity of the molecule based on just having a band. But if you're working in some sort of in vitro system where you're working with purified components or partially purified components, you're pretty sure which band it is. Verify that it actually is the band. So the best way to do this is with like mass spectrometry, where you actually cut out that band and send it to the mass spectrometry, where basically they cut it up into little pieces and send those pieces fly to a machine and identify the sequence of those pieces and compare them to a database. So basically they're able to verify the identity of those products. I've seen, or you can at least to the minimum do a Western blot to make sure it's your protein of interest. I've seen it happen where colleagues have been thinking that they're purifying a protein, and really they're purifying a contaminant in the purification prep, like such as a chaperone, and not actually the protein that they think they're going after. So make sure you verify the protein that you're going after, but once you get a sense that you really are looking at the right band, you can tell things about what else is hanging out with that band. But remember that there could be things hanging out with the band that you can't see because there's not enough of them, because they're overlapping in size, etc., etc. Speaking of not enough of them, now let's talk about the quantity. So the stronger the bands in general, the higher the quantity, especially if you're comparing the same bands, but in like different lanes. So in like different samples, but the same protein that you're looking at. This is because the different proteins will bind dye differently based on their composition and their size. So how these dyes typically work is that they're binding to specific amino acids mostly. So in the case of chromacy blue, it likes to bind to lysine, arginine, histidine, tryptophan, phenylalanine, and tyrosine. So basically, there's various amino acids that it's biased to bind towards. Because different proteins have different combinations of amino acids, they'll have more or less of those ones that Bradford prefers to bind to, or that chromacy prefers to bind to. So different proteins, even if they have the same size, if they have the same molecular weight, they might bind differently. So you can have proteins that are binding more or less, and therefore they would show different intensities on a gel, even if you had the same amount. Another thing that influences the intensity you see is going to be the size. Just as with some proteins, they might have more of these letters that can offer more binding opportunities. In general, a longer protein, a bigger protein, is going to have more opportunities for binding than a smaller protein. And so with the smaller protein, you're going to need more copies in order to get the same amount of signal, because you'll need more copies to get the same mass amount. Much more of this in other posts, but you will need to take this into account when considering how strong the bands are going to be, that if you have a littler protein, you can and a bigger protein of the same concentration, and you add like the same number of moles, so the same number of copies of that protein, the smaller one's going to show a fainter signal than the bigger one. So you might see a strong band for a bigger protein and a weak band for a lower protein. And this doesn't mean that there's less copy-wise of the little protein. It could just be that the little protein has less for the dye to bind to. So be cautious when trying to compare the intensity of bands for bigger things and the intensity for smaller things. But what you can compare is the intensity of the same band under different conditions, such as when you're trying to assess induction efficiency. So basically the idea with induction is that we can get cells to make a protein on demand. A common way to do this is with this T7 system in bacteria, which I've talked about a lot more in another post. But when you add this chemical called IPG, it's going to induce the production of the protein. So it's going to get the cells to start making a specific protein. And so you can run samples with before induction and after induction, or with different amounts of IPTG or a different time course. And you should see the appearance of a band of interest for your protein. And so again, you can't tell the identity of all these bands. So here's like a pre-induction, post-induction, pre-induction, post-induction, pre-induction, post-induction of a different protein. So it's basically testing different things. And you can see that if you look at the pre-induction and the post-induction, you have a band appear here that wasn't present here. And this is likely the product that you are inducing. With bacterial induction, you can actually get the bacteria to start spending like 50% of its energy, or like 50% of the protein it makes. I can't remember exactly which, but for making a protein of interest. And so you can see that you can get a nice fat band if you get good induction. This can be helpful to do before you try to purify something, is to actually see whether it's actually getting induced and what the conditions are for best induction. And so you can see that you're getting good induction here. You appear to be getting induction of a product here, but likely not as strong. And here you can see that if over time or higher concentration, you're able to get more and more of this induced product, which you can then purify out. And since we're comparing between the same protein over time, or in different samples, we can then compare based on the strength of the bands. Okay. We're also going to have that bias for nucleic acids, where the longer things are going to show up stronger than the shorter things, because of just having a longer length and more to bind to. So those fluorescent stains are typically going to be binding just generically to those nucleic acids, but if you have a longer nucleic acid, there's more space for it to bind to. So that is what you can learn from a gel. You can learn about the relative size, purity, and quantity, taking into account all those caveats, such as the fact that bands can overlap, small things are going to show up fainter than bigger things, and the composition of the thing can cause the dye to run, cause the composition of the molecule can both cause the molecule to run slightly different in terms of its speed and therefore how like big it appears based on compared to the latter. And it can also impact the dye binding, and thus how strong the signal looks. But gels can be helpful because they're telling you about like everything that's there, taking into account those caveats. They can't tell you about the identity though. A blot can tell you about the identity, but it can't tell you about what else is in the sample. So if we did a chromatography staining, we could see all the proteins that were present. We did a Western probing for a specific protein. Now we can see just the protein that was present. Yeah, so this is a different gel, but you can see that different proteins are going to have different amounts of these different products. And you can see then you'll get a band if the thing of interest was there and not if it wasn't. So with the Western blot, you're using antibodies. With the Northern or Southern blot, you're using radio-labeled DNA. And ideally, each of those bands would represent that specific molecule you're probing for was present and nothing more. But that's not always the case. So sometimes, although we saw nice crisp bands here, this is just the ladder, but then you can see that for each of these, you get a nice crisp band. What can happen is that sometimes you get antibodies that are a lot less specific, and so you could get multiple bands, and you might not even know which is the band that you're looking for. So how do you know? One thing that you can do is you can compare it to the ladder, and hopefully the contaminants are going to be a very different size than what you're actually looking for. Another thing you can do is include controls. So if you include a negative control that you know does not have the thing that you're looking for, but has everything else, then any bands that you see shouldn't represent, should be just your background. And so if you have like, say a knockout line, which doesn't make that protein, so the cell line that doesn't have that protein and then the cell line that does, you can then compare. If you have a positive control of a protein, like the purified protein that you're looking for, then that can tell you where the band that you're looking for should be. And again, hopefully you won't have any contaminants that are around the same size. You can also try to see if you can get better antibodies so that are less cross-reactive, so that binds to fewer things. You might also here you have the problem of sensitivity, where your antibodies, if you don't have enough thing present, you're not going to get high enough sensitivity to detect it. But you also have a problem where if you load too much, then you're going to get more of an issue with potentially having background. So it's all a balancing act. Speaking of different like strengths, so different antibodies can have different like cross-reactivities, and they can also have different sensitivities. And so you can't really compare antibodies. You can't compare the strength of the band between different proteins that you're probing for. Similarly to how we saw before, you can kind of compare between the same protein or the same nucleic acid, say if you were doing one of the Northern or Southern, in different samples, because you're comparing kind of apples to apples. But if you're trying to compare between different proteins, say with different antibodies, well, now you're going to not be able to compare directly, because those antibodies could have different binding strengths for the things that they're recognizing. And therefore, you might see a stronger band just because the antibodies are like better, instead of because there's actually more of a specific thing present. When I talk about being able to compare between samples, though, so say to be able to see if there's more of a protein made under certain conditions than under other conditions, you need to make sure that you're loading the same amount, like the same total quantity or the same input amount, the same cell volume and things like that in these different samples. And so often you're doing some sort of loading control where you're also probing for like a housekeeping gene, something like GAPDH or tubulin. That's expressed at relatively constant levels in different cells and under different conditions, in order so that you can kind of normalize out for differences in how much you load, so that you don't think you have a lot more, in a sample because the conditions are different, when really you don't have a lot more in the sample because you loaded a lot more of the total stuff. So I hope this helped you see that gels and blots both have their pluses and minuses. And of course, the blot is coming after a gel. So if you stop at the gel and just do an all protein or all nucleic acid stain, you can get ideas about size, purity, and quantity, but not identity. With a blot, you can tell about identity, but only if you're testing, if you know what identity to test for. So it's kind of like a yes, no, is this specific molecule present? But it can't tell you what else is present. Often what you see is that people will show you both the all protein stain and the blot, or an all nucleic acid stain and the blot, so you can get an idea of what else is present. Sometimes these stainings aren't compatible with the blotting, so you can run the gels separately and stain them separately, or you can use some sort of reversible stain, such as if you do like ponzo stain on your membrane before you actually do the blotting step. So I hope that helped you understand the pluses and minuses of these different methods, as well as some of the caveats, things to keep in mind, that the size, purity, quantity stuff, this is all like in an ideal situation, and you have to take into account the fact that different proteins can overlap and hide one another in your gel. You can't know for sure whether anything is actually what you think you're looking at, and things like that. But these are really important techniques, so don't get like discouraged from these caveats. They're still super duper useful, and for the most part, these rules tend to be very helpful, or these concepts tend to be very helpful in terms of size, purity, quantity. So you want to, especially when you're working with a purified or partially purified protein, you want fewer bands, larger size of those bands, and at a location compared to the latter that's consistent with what you would expect based on their size.